Marguerite, Vantagoli, and I work with Dr. Jonathan Rowland this summer. What I've been trying to do is evaluate the health of the aquatic ecosystem. And to do this, I want to quantify the effects of endocrine disrupting compounds on the fathead minnow, which is my model organism. First, to discuss my topic, I have to go over what the endocrine system is. What it is it's in animals. It's responsible for signaling throughout the body, and the way it does this is by using hormones. Throughout the body, it regulates your growth and development, reproduction, and metabolism, among other things. Here's a general picture of a fish's endocrine system. An example of one of the hormones that is used is insulin. And hormones are used across long distances, and here we can see that the way insulin works is when you take sugar into your system, the cells in your intestine recognize that and transmit a message using hormones, using insulin, other hormones to the pancreas to produce insulin, which then brings the sugar into your cells. And just like anything else, humans can screw this up too. And the way we do this is by endocrine disrupting compounds. What they are is synthetically man-made chemicals that are foreign to an organism's body. They're not natural, they don't belong there. They work in a variety of ways. They can either block naturally occurring hormones and alter the gene expression, or enhance these hormones and pathways. One example that I plan to work on in the fall is by using a phenylestradiol. What it is a synthetically made compound, and it's the main component in most birth control pills. So based on that, we know that it must act as an estrogen, which is shown on the screen behind me on the right. And we can see that these molecules are almost identical. And the only difference is that on the phenylestradiol, you add a phenyl group. So this acts just like an estrogen in your body. The way we want to do this is by developing a biomarker. So what a biomarker is it's a physiological response that can be measured. It's a response to any trigger. A simple example to grasp this is the idea of plant growth. You can measure plant growth, and theoretically, the more fertilizer you add to that plant, the more it's going to grow, and that can be measured. The goal of this project is to detect these compounds before adverse effects occur. So we want to find out that these endocrine disrupting compounds, specifically estrogen-like ones, are in the water before they affect populations by causing either population die-off or impairing the reproductive capabilities. The way we're going to do this is through RNA. And for basic biology, DNA is like the genetic library of a cell. It houses all your genetic information, it's hereditary. RNA is copied from this to make all the proteins that Dave was talking about that actually make you who you are. So RNA is like the middleman. We can quantify this, and we'll go into that later. So why do we care? We can see, obviously, that this fish has frog has 3 paired legs. This isn't normal, and this is actually caused by endocrine disrupting compounds. They've been detected in water, in drinking water, in industrialized nations across the world. There's even studies correlating them to breast cancer rates on Cape Cod, being in the drinking water there. These are pictures of the fish that we're using. On the top, you can see that that's our larva, and the fish egg is on the right. And on the bottom left is an adult female fathead minnow, and the right is a male. We finally got them to mature. That was a lot of work in and of itself, but you can see that the males are a lot darker than the females, and that's how we differentiate them. So what my research question is whether or not we can monitor the presence of these endocrine disrupting compounds in the environment, and specifically the aquatic environment, by using these fish. Basically what we do is we move from the fish we homogenize the fish, isolate RNA, perform a cDNA synthesis, and a polymerase chain reaction of the target genes. For an RNA isolation, what we do is we euthanize a fish, homogenize the sample into a vial, and this is what we have here on the left. There's all the components of living matter. You have your lipids, your proteins, your DNA, your RNA, and you also have your carbohydrates. So somehow we have to get from all of this stuff to RNA. And to do this, we perform a total RNA extraction. We actually tried two methods. One is RNA aqueous. It's a kit that you purchase from Ambion. The benefit is it's not as toxic as like a Stat-60, which is another method that we use because it uses a lot more like abrasive chemicals. We also tried the Stat-60, which is more cost effective, and we've stuck with this because it gives us almost 100 times more RNA than the much more expensive RNA aqueous kit. Here we can see, this is just results of these kits, and this is how we determined which one worked as well. On the left we can see that I ran a gel electrophoresis of both of these samples. The RNA aqueous kit doesn't show up at all, and the Stat-60 kit, it's probably really hard to see on there, but it shows two distinct bands, which shows that there is in fact RNA there. There's the 28S RNA and 18S RNA, and the reason these show up most predominantly is that they're in every cell in your body in large concentrations. So once we have the RNA, we perform a reverse transcription reaction, and this is where you go from mRNA to DNA. Normally in a cell you go from DNA to RNA, but this is going backwards. Then we eventually use the cDNA, the complementary DNA, in a PCR reaction, which is called a polymerase chain reaction. Basically what the goal of this is to amplify a target gene, one of the biomarkers that we're going to be looking at. And the way you do this is you heat it up, the DNA splits apart, and it just through several cycles you replicate this. And from even one copy of DNA, because it doubles every time, and you do 30 to 40 cycles, you get 2 billion copies of that target gene. From here we perform a quantitative PCR reaction. The goal of this is really how our research works. It quantifies gene expression, and it works just like PCR, as the name suggests, but it adds fluorescent dye called cybergreen to double-stranded DNA. As this shows on the curve, on the bottom axis is the cycles as the PCR reaction progresses. So like I said, we could do, depending on the reaction, 30 to 40 cycles. And as the reaction progresses, you can see an exponential increase in the amount of DNA that you're producing. To determine what the starting concentration of our DNA was, you draw a threshold line, which is the red line, and then you drop that down to the axis, and you can quantify how much you started with relative to the other genes or the other concentrations. We can see at 23.5, that's when that first curve began to rise. And compared with 39, we know that there was more of the 23.5 in the tube to start with because it rose faster. It took less cycles to get to that product, to get to that line. From this, we run a standard curve. The sample gene that I'm using right now is estrogen receptor beta. From this, it just shows us that our DNA is reproducing in a linear fashion. It means it is doubling every single time. We also run a melt curve analysis on the qPCR machine. And from this, we can determine the purity of our sample. The problem with using primers is sometimes they'll bind to themselves instead of binding to the DNA and amplifying the target gene. On this one here, we can see a single peak around 85 degrees. And that's promising because it shows that the sample is pretty pure. It's only making one sample, one product, I'm sorry. This slide here, it happens to be androgen receptor. We can see that there's two distinct groupings. On the left, there's two curves that show up completely different from the two on the right. And this caused us to have to throw out these primers because they're not going to help us at all. They're not making a continuous product. Even though the standard curve data might have looked good, we can't make two products because you can't quantify that. Through the PCR, we've had a few successful genes. Some of them are shown here. We have 18S RNA, which is a good housekeeping gene. So when we expose our fish to endocrine disruptors, theoretically this should be, because we're using estrogens, this should stay around the same. It'll be a good marking point. We also have CYP3A, which is a gene that breaks down pharmaceuticals, which is ethinyl estradiol. Beta-actin 2 on the bottom left is also a good gene because it's another housekeeping gene, just like 18S RNA. And we also have vitilogenin. This is really important because vitilogenin is a protein that's expressed only in female fish. It's an egg yolk protein, so it's only when they're about to lay eggs. Males don't express it. It's in their DNA, but it's not expressed. If we can see that male fish are expressing this vitilogenin protein, we know that they have been exposed to something that acts as an estrogen. So it's a good promising gene. So here are some of the other genes. We also had success with both estrogen receptor alpha and estrogen receptor beta, and as it said, vitilogenin. Some of the other genes that weren't successful are listed here. One was one type of beta-actin and another type of vitilogenin, which is a different primer set. But we don't really need to go back for those and double check them. Aromatase and CYP1A, we're going to go back and make a new set of primers for in the fall and test those again, along with several more genes. The future work for this project is we're going to be performing a plasmid verification. This is just to make sure that we're going to sequence the genes that we've PCR'd, just to make sure we're targeting the correct gene. Like I said, we're going to develop primers, some more additional sets of primers, and also run the exposures to ethinylestradiol. And we hope to determine the feasibility of these genes as potential biomarkers. I wanted to thank the ATP program. I never really noticed, or I've never really thought of how much it really costs to do molecular biology. And this is, animal care was about $3,600. Molecular biology is actually $26,000. So